fermentation. 11C, fermentation. Fermentation. The yeast cells are fermenting the sucrose, the sugar. Of course, you know, they will break it down to alcohol and carbon dioxide. Which of the following can cause shrinkage of living cells? That's number 12. The correct option here is D, hypertonic solution. Hypertonic solution. When a cell is surrounded by a solution that has more solutes, the cell will start losing water to that solution. Hypertonic solution has more solutes than the cell. It has more solutes than the cell, so water moves out of the cell to that solution, because water moves from dilute solution to more concentrated solution. Water moves from dilute solution to more concentrated solution. So that is hypertonic solution. So it has more solute in the cell. So it will cause the cell to lose water. So the correct option here is hypertonic solution. Isotonic solution has the same amount of free water molecules as the cell, or the same amount of solute, so there is no net movement of water molecules. For hypotonic solution, it has more free water molecules than the cell. So when a cell is surrounded by hypotonic solution, the cell gains water instead and becomes turgid. Question 13. Which of the following is true of leukocytes? Leukocytes are white. Leukocytes are white blood cells. They are white blood cells. So they are large and nucleated. Which of the following is true of leukocytes? They are large and nucleated. Okay. There are two main types that we study here at O level: the lymphocytes and the phagocytes. Okay, so when you look at phagocytes, they have this irregular shaped nucleus. So the correct option here is C. The conversion of a nutrient into molecule in the body of a consumer is referred to as assimilation. Is referred to as assimilation. Assimilation. That's for instance, you eat a carbohydrate food, is converted to or broken down to glucose, is carried to the liver, and the liver converts it to the excess glucose to glycogen. That's an example of assimilation. When the body makes use of the nutrients, that's assimilation. The ability of living organisms to detect and respond to changes in the environment is known as irritability. That's B. Irritability is also called sensitivity. Sensitivity. Okay, so organisms are able to detect and respond to changes in their environment. Okay, the chameleon can change the color of the body depending on the color of the environment. So the correct option here is irritability. In mammals, the exchange of nutrients and metabolic products occurs. Okay, we take lymph option D here as the correct option because, well, if you look at the capillary bed, nutrients are exchanged between the tissue fluid and the cells. Of course, you know, the plasma that seeps out of the capillary also come with the nutrients from the blood. So that is what forms the tissue fluid. So materials are exchanged between the tissue fluid, which is formed from plasma from the capillaries, and then the cells. An example of an endospermous seed, that's a seed with endosperm, is A, the maize grain. The maize grain is endospermous, contains endosperm. Of course, you know, after the double fertilization in flowering plants, a structure called endosperm is formed. In monocots especially, the endosperm is still there after the fruit or the seed is developed. So it's a food storage structure in the seed. Okay, so you still find that in the maize grain. So which of the following is correctly matched with the organism that it exhibits? So you have I here, parasitism to sundew, autotrophism to amoeba, saprophytism to alga, heterotrophism to agama. Which of the following is correctly matched with the organism that it exhibits? The agama lizard is of course heterotrophic. It feeds on already made food. That should be our correct option here, C. In which of the test tubes will glucose be detected after complete hydrolysis? Test tube 1 contains cane sugar, that's sucrose, and water. Test tube 2 contains cane sugar and diluted acid. Cane tray contains test tube 3 contains cane sugar and degrading enzyme. Okay, so the correct option here is 2 and 3. That's option B. Option B, 2 and 3, because the sucrose will be degraded by the acid and can also be degraded by the enzyme. Okay, it can also be degraded by the enzyme. Of course, you know, The enzyme sucrase breaks down sucrose to give you glucose and fructose. So the test tube containing the sugar and enzyme, there will be hydrolysis there to be degraded. The one with sugar and dilute acid, there will also be hydrolysis there to be degraded. The enzyme involved in the hydrolysis, of course, is sucrase. Sucrase degrades sucrose to give you glucose and fructose.